Hi, my name is Matina, Prapa. I'm a clinical geneticist. I work in London at few centres, St George's and the Royal Brompton Hospital. I've been a geneticist for 3 years now as a consultant, and I stumbled into the world of pulmonary hypertension as a trainee in clinical genetics in Cambridge, and that has created a big connection for myself in terms of TBX for life, because I studied the association between the genetics of TBX4 and the manifestations of the condition itself.
So I'm Olivier Danhaive. I'm Belgian. I'm much older. I've been 30 years plus in neonatology. I'm a neonatologist, trained in Belgium and in the US, and worked in Italy, in the US, and in Belgium. Now, for the past 5 years, I've been the director of neonatal intensive care in Brussels at the Catholic University of Louvain. So that's my work. I always was intrigued clinically by those infants or children or newborns with rare diseases, with special conditions that we would not necessarily understand. They were really a trigger to my scientific curiosity. And I came into getting interested in lung disorders, especially because they were pretty challenging to manage. We started to explore the genetics of lung disorders and understand that there's many types. Some are problems of development, some the lung lacks a piece or has some malformation, and all of these connects to certain genes and certain mechanisms. And I came into one of the first patients who was affected with a mutation of the gene TBX4, which we didn't know at that time because it hadn't been really described as a disease. That really hit my memory, and it triggered an interest in collecting and finding among my colleagues and collaborators more and more cases in order to get a better understanding of this disease.
It was serendipitous. So I was introduced. I don't remember how it came about, but I became a member of TBX for Life when it was in its infancy. And they were looking for volunteers to take on management of the working groups. And I just raised my hand and said, I'm happy to do it because no one else was really volunteering. We was quite junior, but I thought maybe that's my way into a leadership role as well. And the team is lovely. But to be honest, I was struggling with the admin workload and trying to manage the whole thing on my own. And we worked quite nicely with Olivier. And at some point, I just asked him, why don't you join me? And it's very nice synergy because we're coming from different fields, different countries, different levels of experience, but it works quite well, I would say. And so that's how we met each other. It was online, but now we've been through a few meetings. And I met you in Cambridge as well, in person, yes.
Right. Yes. Well, I came late in the group. I just joined a couple of years ago because some of the people I had collaborated with in the US became members of TBX for Life, in particular people from the PPHNet group, Steve Abman, and they said, Olivier, by the way, you wrote that paper that we're all talking about because it's kind of describe the phenotype of the newborns. Why don't you join? And so I was really happy to come. And that's how I came across Matina, which I had met before, actually, but we started working in her working group. And as you said, at some point, we kind of share a little bit the driving of this group, trying to get things moving, advancing knowledge and science, because that's what we're here for.
I would say we're very international. A lot of centers, mostly across Europe and the States. And we have two working arms at the moment. So it's the clinical arm and the basic science arm. So the people who do the lab work and the people who are more patient-facing. But we do speak to each other a lot. We are both working towards particular goals. And we can talk a little bit about what we have been doing. One of the things that we stumbled upon and understood is that people struggle with they get a diagnosis, where do we go from here? So essentially, the number one thing we want to do is to draft some guidelines or recommendations so that you can pass on to your physician and say, I've got this rare condition. This is what we should be doing moving forward. So that's our number one goal at the moment. But there are lots of other things that we want to address. One is we want to complete a natural history study because we know there's huge variability even within families with the exact same gene change. And that might give us a better idea for future clinical trials in terms of modifying genetic factors, for example, why a father is unaffected, but the child is severely affected, or vice versa, and take it from there. But these are our two main focuses. From the basic science perspective, there's a lot going on with mouse models, cell lines, etc., which complement the clinical work quite nicely.
One pitfall for the families and for ourselves as well is the journey that it takes from when the disease manifests itself to when you get a diagnosis or cause. In my feeling, in my perception, and talking to so many families, that's so hard to have questions in your head about a son or a daughter or a family member. You see that something is serious, something is happening, something is progressing, and you don't know what it is, and maybe the doctor doesn't exactly know what it is either. That's really one of the biggest challenges in rare diseases, in particular with pulmonary hypertension. Because pulmonary hypertension is a rare disease, and there's not that many people who know about it and to know how to really untangle the genetics, the clinics, the treatments. And also, the second challenge with TBX4 is that it's a disease that has so many shapes and facets. You can get, sometimes you can get an orthopedic disorder, kind of toe problem or knee problem, and be perfectly fine. Others have pulmonary hypertension, which is very severe. It's a big burden in terms of health. And that can happen at several different ages, different periods of life. And for each of these manifestations, each of these ages, you have different doctors, different specialists who don't necessarily talk to each other. That may be the biggest step that's been brought by something like TBX for Life. Because now I would say, independently of TBX4, if I had a newborn in my unit with a challenging disorder I don't understand, I have a community of collaborators I can call immediately. And we have a network, and we exchange knowledge, and I know exactly who knows what and who can help me with what. And that's a fantastic achievement.
Patients and families rarely say no to genetic testing, but obviously when you do consent them and explain why we want to do the test, you have to explain the pitfalls as well. And there are several negatives or potential pitfalls. One is the results of the test. We always say to families are not black or white necessarily. It could be a grey result, where the so-called variants of ascending significance, which were burdened with in genetics. That can be difficult for the family to cope with and understand what it means. Sometimes it's the sense of guilt, so especially if you find a variant, by variant I mean a gene change that was inherited by a parent, so there's some guilt. We always say to parents, look, we all pass on things to our children, and it's a matter of bad and good luck, really, and sometimes you just don't know, but the silver lining is you can do something about it now. And then, of course, the upside is that families, they find it so helpful to actually have a name or be given a diagnosis. Sometimes it's very difficult because they say, okay, now what? Is there a cure? Is there a gene therapy? And I think we're moving towards that slowly, but there's still lots of things you can do in the meantime to try and optimize treatment.
First of all, I would say in the past 20 years, the number of disorders for which a causative gene has been identified has been multiplied by 20, 30. I mean, it's really been a boom due to the natural progress of medicine, also by the incredible progresses of technology. When I started, you would sequence a gene like you would read a book, like you would literally, there was a machine that would read bead by bead by bead each component of one gene. Now you just blast the whole genome and you get a printout of what's in. So there's been totally an acceleration both on diagnostic and research on that field. And then genes are being repaired and replaced now. I mean, it happens. It really happens. It's still a dawn. I mean, it's still the beginning, but you have disorders that were otherwise lethal and they're not lethal anymore. So that you can really cure genetic disorder. When you think about it, it's kind of magic.
It's a very exciting field, massively exploding in terms of massive parallel sequencing, which is what Olivier was talking about. You can sequence someone's entire DNA within a space of a day. Problem is you're left with so much data that you can't necessarily interpret. You have to be quite cautious in terms of how you read this genetic data and how you interpret it. As you know, there's been massive explosion of direct-to-consumer testing as well, which again is not necessarily helpful. And I think this is where I see a little bit of, not of mistrust, but misinformation, I would say, from families that they go and have 23andMe or something else done, and then they come to you and they expect to get some answers for genetics that we know that don't really contribute to a condition like early onset Alzheimer's or hypertension, for example. But for the ones that we know there is a good association between a single gene change and the condition, we're on the frontier of gene therapies. And we have the tools, just a matter of how to deliver, how expensive and how fast it is to actually develop those therapies and get them to to the patient. I can't speak for the US, although I know that from experience there are lots of people who have access to genetic testing, it seems like. So I can speak for the UK. It's one of the frontiers of the so-called whole genome sequencing, which is what I mentioned, just reading the whole DNA, so the entire genome in the national healthcare system, so for free for the patient, for several indications. So I think what the desire is for the UK at least is for every single patient who comes to the national healthcare system is to have their whole genome sequenced. Obviously, this is for research purposes as well, not just for diagnostic purposes, because they want to build a library where we can carry on studying the genetics of people and try and unmask new conditions as well. I don't know about Belgium, but you're quite advanced as well, aren't you?
Well, Belgium, it's not such an ambitious nationwide program, but we do have labs and university and tools. Belgium is really fragmented as a little country, but that's the way it is for pretty much everything, from politics to medicine to research. But I would say in terms of cost, it's now totally in the same magnitude as any test. In terms of sheer costs, technical cost, I mean, getting an exome sequencing, it's a little bit more expensive than getting an MRI, so it's just mainstream medicine.
So with Saturn, we tried to formulate a roadmap for the next 3 and 5 years, which is a timeline of what we want to be achieving, what's realistic, what's unrealistic, where will the funding might be coming from, etc. So I think this is where we are at the moment. Obviously, Anton, who is the CEO, Anton Morgan, the CEO and founding member of TBX for Life, he wants a cure, and that's where we are supporting him, and we're putting all of our money in terms of efforts. How close are we to that? Not very close, but there's amazing basic science research going on, approaching it from right and left, not just on TBX4, but generally for pulmonary hypertension. And I think we will be seeing more in the next couple of years.
Sometimes the gap between science and actual patient treatment, the gap may be wide, and because there's so much regulations. It's not only that you need to invent the therapy, but that you need to validate it. You need to sort it out, to go through a whole series of approval and tests, and that takes years, if not decades sometimes. So that can be pretty discouraging. If you find out that a compound, like a drug, like a molecule, could be a good candidate to help people with TBX4, let's say. So you really need to go getting this laboratory compound into a pill or an IV drug. That's going to take 5 to 10 years. Instead, if you test existing compounds very massively, you test like 10,000 compounds on a single chip, for example, and you find some existing drugs that are used in cancer or in cardiac disease or whatever are actually active. This is a shortcut because it already has an identity. It's already been tested. It's already used in human. You can actually buy it. And these are incredible shortcuts. And that's a little bit the kind of approach we're looking into at this stage, as a transition to real genetic cure, which will clearly take more time. So I think there's very exciting things, not only for the beauty of science, but like literally for people who maybe are already born or are already affected now. And if not, really the ones who are coming in the coming days or months or years, I think we're close to be able to offer some new things.
TBX4 is quite a tricky gene because it's what we call a transcription factor, so it switches on and off several of other genes. And that's where the tricky bit comes from. It's just not just about replacing it, it's about what it does and at what point in time. It should be happening in utero when the baby is developing in the womb. Is it too late if you try and do something 5 years down the line, etc.? These answers we don't have yet, but that's what people are trying to get to.
What is striking here is that every bit and piece of information, everything that one researcher does, comes together for the final purpose. Like Matina was mentioning, doing the natural history project. So it's really describing what happens, put all the individuals with the TBX4 gene mutation, which can go through a terribly sick little baby to a 40-year-old who's perfectly healthy is just being tested because he has a family member who's a carrier. Having all those listed together, we can really kind of get a sense of the disease. And that's extremely helpful, not only to design future therapies, but also to understand what's going on, how the gene and its mutation impact health or not. So I think the fact of putting people from so different horizons is really what makes it stimulating and great. Thanks for listening. My name is Olivier Danhaive.
And I'm Matina Prapa, and we're aware that our patients are rare.